Hello everybody. Last time we had seen amino acids. Each amino acid contains a distinctive R group. We had considered the commonest occurring primary amino acids or the standard amino acids, and we had seen that every amino acid has got an R group, which is also called the functional group. So today we are going to see special roles which are performed by these R groups or the functional groups. So R group we had seen varies. There are some sulfur containing R groups, hydroxyl group containing. There are certain which are dicarboxylic. There are two carboxyl group, two amino group, etc. etc. So we had seen all that last time. Now today we are going to see special functions performed by some R groups. Now for example, cysteine, a sulfur containing amino acid, it is involved in cross-linking of the adjacent polypeptide chain. Now for example, this is one cysteine, this is another SH group. You can see here by elimination of hydrogen they can form a dimeric structure. So this dimeric structure is called cysteine. So what is the role of this? What is the purpose of this? Formation of a covalent linkage stabilizes the structure. It provides strength to the structure. So appropriately placed cysteine, they can form disulfide linkage, which is important for stabilizing the polypeptide structure. And an important point that they occur intra-chain also, within the same chain also, they can be present. So cysteine is the one sulfur containing. Another one we are going to talk is methionine. Methionine agar aap dekho, it has got a not a terminal SH group, but SH group ke baad there is one methyl group also. And methionine in its activated state you will learn later on, that is called S-adenosyl methionine. So this is capable of donating the methyl group for synthesis of a number of important compounds. Lecithin, a phospholipid, betaine, a lipotropic factor, checks progression of fatty liver, carnitine involved in fatty acid oxidation. All these three are methyl-rich compounds, and donor of the methyl group is methionine in its activated state, SAM, S-adenosyl methionine. Now we come to hydroxyl group containing amino acid, serine and threonine. They play important role in regulation of enzyme activity. तो enzyme activity regulation का मतलब क्या हो गया? Metabolic pathway का regulation. Just consider this is a protein, an enzyme protein, and this is a hydroxyl group of a serine residue. This hydroxyl group can be phosphorylated. Phosphorylation can cause activation of this enzyme. An example we are going to see here, glycogen phosphorylase. It's an important enzyme for glycogen degradation. After it is phosphorylated, it becomes active. And another enzyme of this pathway, glycogen phosphorylase kinase, this is also activated by phosphorylation. So some enzymes are activated by phosphorylation, some are deactivated by phosphorylation. But the important point here is that serine ka hydroxyl group, it acts as a important regulator of the enzyme activity, reversible enzyme activity. Now we come to the next one, proline. You can see here proline. It has got a distinctive feature. The R group is cyclic in nature. R group is this much, is cyclic in nature and it includes amino group also. तो इसलिए proline को it is sometimes called amino acid. So it contains a pyrrolidine ring structure. Because of this peculiar property, a peculiar result comes into picture. Proline जहां भी होता है, for example, इस polypeptide chain में, supposing if proline is here, proline will introduce a sharp bend in a polypeptide chain. You have got a polypeptide chain, आप यहाँ पर proline introduce कर दीजिए, immediately at this point it will bend. So proline, that is why it is also called a helix breaker. तो जो कुछ polypeptides which are very tightly, जिनका some polypeptides which are very sharp bends etc. वहां पर proline होता है. One example is collagen. Collagen, it consists of 3 polypeptide chains which are tightly interwound around one another. And the sharp bend देने के लिए proline का होना जरूरी है. Glycine also plays an indirect role in collagen. क्या करते हैं glycine? As we said that the 3 chains are very tightly interwound around one another. So very little space is available in the interior. And इतने कम space के अंदर सिर्फ glycine accommodate हो सकती है because it has got a smallest R group, that is H. So we are going to see in a clinical example how important this indirect role of glycine is. So now we come to aliphatic non-polar R groups. These are the amino acid, alanine, leucine, non-polar aliphatic R groups are there. इनका भी एक indirect role है which we are going to see. अब ये देखिए, the chains, polypeptide chains, they sometimes in the globular proteins, they tightly, fold. Extensive folding is there. Extensive folding होने के बाद क्या होगा? The hydrophilic amino acids will remain on the exterior. But the hydrophobic amino acids, they will move away from the water. जितना दूर जा सकते हैं जाएंगे. As a result, they will be buried in the interior. तो इस टाइप के जो ग्लोबुलर प्रोटीन्स हैं, उनका एक हाइड्रोफोबिक कोर बन जाता है because the hydrophobic amino acid, they cluster together to form this core within the protein. और इस कोर के अंदर water is totally excluded and these hydrophobic amino acids के R group, they associate with one another in what is called hydrophobic interaction. So hydrophobic interaction along with some other forces, they are the strongest driving forces which keep this three-dimensional conformation intact. So forces stabilizing the three-dimensional conformation, 3D conformation of the globular protein. Most important is hydrophobic interaction, and the main player here are chief actors here are the aliphatic non-polar amino acid. Van der Waals forces, you must be knowing, positive nucleus of one molecule interact with electron cloud of another, and some other forces are also there. अब रह गए दो तीन रह गए. asparagine. Asparagine you can see here, इसका CONH group है. It is formed from dicarboxylic aspartate. तो इसका CONH group का क्या रोल है? I have written GP here. GP means glycoprotein. Asparagine, यहाँ पर CONH, this acts as an attachment site for oligosaccharides. तो ग्लाइकोप्रोटीन होते क्या है? प्रोटीन जिसके ऊपर एलोगोसैकेराइड अटैच होते हैं, और एस्पार्जीन प्रोवाइड्स द अटैचमेंट साइट फॉर oligosaccharides. Histidine, this we will do in the next class. It is the only amino acid which is able to act as a buffer at the body pH. और lysine, last one probably, lysine का क्या है? You can see here, lysine has got a long CH2, 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 NH2. It has got a very long R group. And this R group is capable of binding with certain coenzymes. Coenzymes like biotin, pyridoxal phosphate. So this provides attachment site for certain coenzymes. When you do metabolism, you will understand more about them. So we have done these amino acids and we have seen roles of a few. Just a clinical corner, हमने देखा था कि glycine is the only amino acid which is accommodated in interior of collagen and provides strength to it. Supposing if glycine were to be replaced by a bulkier amino acid, cysteine, फिर क्या होगा? कोलेजन का इंटैक्ट स्ट्रक्चर, उसकी स्ट्रेंथ डिसरप्ट हो जाएगी, it will become fragile. The disease is called osteogenesis imperfecta, brittle bone disease resulting due to incorporation of bulkier amino acid because of point mutation. इससे फिर क्या होगा? Bone fractures होंगे. Cystinuria, यहाँ पर क्या है? The amino acid cysteine, it is normally reabsorbed. The transport protein undergoes some defect. As a result, cystinuria, cysteine will be eliminated in the urine where it will form stones. So renal stones will develop in this condition. To summarize, what is cysteine? For holding polypeptide chains together or two different regions of the same chain. Methionine, donor of methyl group, thereby synthesizing various compounds. Serine, threonine, regulation of enzyme activities. Non-polar aliphatic, we have seen hydrophobic core of globular protein. Or histidine, it has imidazole group for buffering activity, asparagine for glycoproteins attachment, lysine for coenzyme attachment. So we have done about the standard amino acid, but our body also has got some non-standard amino acid. For example, hydroxylysine, phosphoserine. क्या फर्क है? इनका जेनेटिक कोड नहीं होता. All amino acids in our body are L amino acid, D amino acids only in bacteria. Some non-standard amino acids, I just want to tell that more than 300 amino acids are present in our body. But why we were obsessed? We have been obsessed about those 20 because they are genetically encoded. तो ये फ्री फॉर्म में भी कुछ नॉन स्टैंडर्ड होते हैं. फॉर एग्जांपल ornithine, citrulline, you will learn about them later on. Lastly, glycine is a neurotransmitter. Glutamate is also neurotransmitter. Glutamate, I think it is excitatory transmitter, and glycine is inhibitory. Please check it. So we finish with this class about the R groups of amino acid. Next, we will be doing about, this is important, acid-base properties and buffering action of the histidine. Till then, goodbye. Thank you so much.